Buenas noches, estimados amigos, estimados colegas, médicos, tecnólogos médicos y colaboradores. Bienvenidos a nuestro primer webinar de PositronPharma. Mi nombre es Vasco Kramer, yo soy el encargado de investigación y desarrollo en PositronPharma. Yo voy a ser su anfitrión hoy día guiándoles por este webinar. El objetivo de este evento es introducirles un nuevo producto nuestro que se llama FAN. FAN es una abreviación por un nuevo radiofármaco que se llama fluor-alf-nota-octreotide. Nosotros trabajamos en este proyecto hace 2 o 3 años aproximadamente porque nosotros creemos que este radiofármaco va a mejorar significativamente el acceso a imágenes PET de tumores neuroendocrinos a pacientes en todo Chile, no solo en Santiago. Nosotros también creemos que este nuevo radiofármaco va a ser igual, si no superior al estándar actual que tenemos, el galio dotatate, y eso creo que vamos a ver en las presentaciones científicas. Pero antes de que vamos a esto, yo voy a pasar la palabra a Raúl Carrillo, gerente general de PositronPharma, quien va a dar unas palabras de bienvenida. Raúl.
Muchas gracias, Vasco. Bueno, muchas gracias a todos por conectarse, los que están desde sus casas, los que están acá presencialmente. Vamos acá. Bueno, estimadas amigas y amigos, junto con agradecer la presencia, le damos la más cordial bienvenida y los invitamos a ser protagonistas de este evento, que estamos seguros dará el puntapié inicial a muchos más. Compartimos un espacio desafiante que nos moviliza día a día en la búsqueda de nuevas soluciones para los que están en el centro de estos esfuerzos, los pacientes. Según el Observatorio Global, en el 2018 el cáncer pasó a ser la primera o segunda causa de muerte a nivel mundial, superando a las enfermedades cardiovasculares. Chile no se exime de esta realidad, pues en el 2019 se posicionó como la primera causa de mortalidad. En la actualidad, el cáncer sigue siendo la principal causa de muerte en nuestro país, alcanzando la cifra de 28.400 personas. La Agencia Internacional del Cáncer estimó que para el año 2040 en nuestro país los decesos causados por esta enfermedad llegarán a ser más de 55.000. La incorporación de nuevos dispositivos, drogas, protocolos, inteligencia artificial, sistemas de información y, por supuesto, radiofármacos, entre otros, no sería posible si no tenemos una permanente ambición por responder a preguntas y resolver problemas a través del trabajo en conjunto con las áreas clínicas, en la recopilación y análisis de información, pruebas y diseño para la posterior generación de productos. En PositronPharma estamos convencidos de esto y lo hemos estado demostrando durante todos los más de 13 años de experiencia, desde sus inicios con el doctor Horacio Amaral a su cabeza y el compromiso de un tremendo equipo de trabajo, siendo pioneros en la generación de espacios de investigación y colocando a disposición de la industria productos que no tienen otra finalidad que mejorar diagnósticos, tratamientos más asertivos, es decir, una mejor opción para nuestros pacientes. Sean una vez más bienvenidos y los dejo a continuación con nuestro jefe comercial, Ignacio Postigo.
Muchas gracias, Raúl. Muchas gracias a todos por estar conectados hoy día. Sé que por la hora muchas veces es un poco complicado, ¿verdad?, asistir a una hora determinada, pero bueno, aquí estamos y le doy nuevamente las gracias a todos por conectarse junto a nosotros. Quiero señalarles que, bueno, en recuerdo a la medicina moderna, que ha vivido múltiples avances en los últimos años, que han permitido ampliar el diagnóstico y los tratamientos en la lucha contra el cáncer, y enmarcados en la terapia dirigida, la medicina nuclear desarrolla igualmente su uso terapéutico en oncología, mediante la combinación de diagnóstico y terapia, dando paso a un nuevo paradigma en la lucha contra el cáncer. Las terapias dirigidas con los radiofármacos, sin duda alguna, estas son un gran pilar de la oncología, ya que representan una opción precisa eficaz y sostenible mediante el uso de técnicas moleculares tradicionalmente destinadas al diagnóstico. Estas terapias ya en uso demuestran mejores resultados de supervivencia y mayor calidad de vida en los pacientes, contribuyendo en gran medida a alcanzar los objetivos de medicina de precisión. Y es por ello que PositronPharma les ofrece un abanico amplio de moléculas en nuestro portafolio. Ya sabemos quiénes son, nuestro querido FDG, ¿verdad? Nuestro flúor PSMA, y también están nuestros productos neuro, en los cuales son de desarrollo propio y son la contribución al correcto diagnóstico en este tipo de patología, principalmente Alzheimer y Parkinson, Flor-Betaben, ¿verdad? Y nuestro PRO4. Y por supuesto, el rey de esta noche es nuestro FAM, nuestro Alf Nota Octotrai, como bien lo presentaba Vasco, destinado, ¿verdad?, en el diagnóstico correcto de tumor neuroendocrino primario y metastásicos en los pacientes que lo padecen. Bien, muchas gracias por mi parte, por estar conectados. Los dejo con Vasco Kramer, nuestro investigador jefe de PositronPharma. Gracias a todos.
Bueno, gracias Ignacio. Y con eso vamos a la primera presentación de nuestro programa científico que nos va a dar profesor de Roos de Leuven, Bélgica. Profesor de Roos obtuvo su doctor en medicina desde el Hospital Clínico de la Universidad de Leuven en 2001, lo siento, y en 2002 empezó su estadía como estudiante PhD en la misma unidad. Durante su PhD, él también pasó un tiempo de 2 años en la UCLA y Stanford junto con Sanjeev Gambier en el laboratorio de imágenes multimodales genéticas. Desde 2004 hizo diferentes proyectos de investigación clínica en la División de Medicina Nuclear en Leuven y obtuvo su PhD en 2007. Desde 2006 hasta 2008 también resumió su residencia, que cumplió en 2008. Profesor de Roos fue apuntado profesor asistente en la KU Leuven en 2009, profesor asociado en 2012 y full professor en 2021. Profesor de Roos dirigió 9 tesis doctorales durante su carrera, publicó más que 266 publicaciones peer-reviewed y, más importante, fue el investigador principal del estudio clínico fase 2 y 3, investigando este compuesto alpha-nota-octreotide. Profesor de Roos nos va a presentar su charla con el título Clinical Development and Results from a Prospective Multicenter Study. Alpha-nota-octreotide outperforms gallium-68 dotatate PET in NET cancer patients. Professor de Roos, the floor is yours.
Dear colleagues, I'm Christophe de Roos, a nuclear medicine physician from Leuven. I thank the organizers of this webinar to invite me, and I'm going to present you some results that we have obtained studying aluminum fluoride nitrooctreotide as a novel ligand for the somatostatin receptor. These are my disclosures. Now, you all know that PET tracers consist of different chemical moieties, first one being a vector molecule, which is responsible for certain interaction with a molecular target, which can be a ligand that binds to a receptor or a substrate for a transporter or an enzyme. And to this vector molecule, either to a linker, a chelator, or a covalent bond, a radionuclide is attached, and this radionuclide will emit emission that can be detected by our PET cameras. A very interesting target is the somatostatin receptor, which is overexpressed in neuroendocrine tumor patients. It's a 7-transmembrane G-coupled receptor. There are 5 human subtypes that have been described. The function of this receptor is to diminish secretions, cell growth, and to increase apoptosis. And interestingly, it will internalize upon binding with an agonist ligand, which is a very interesting strategy, of course, to smuggle radioactivity within cancer cells. It's present at the cell surface at a high density, as we can see from this immunohistochemical staining. There have been several generations of diagnostic SSTR radiopharmaceuticals that have been developed by combining different radionuclides with different somatostatin analogs with using different chelators. Some of the most important ones are highlighted on this slide. We have indium 111 pentatriotide, which is of course imaged by scintigraphy or SPECT, and then we have a generation of gallium-68 labeled somatostatin analogs, DOTATOC, DOTATATE, DOTANOC, and then some new kids on the block, copper, DOTATATE, and then the one I will be talking about, ALFOC, as already mentioned, and all of these can of course be imaged by PET. Gallium-68 was the first radiometal that has been consistently used in the clinical setting. First images date from 2001 from the Heidelberg, Hanover group, and in collaboration with Heidelberg. They have shown that they have significantly better uptake compared to indium 111 pentatriotide, which was the gold standard at the time. We started using this in Belgium in 2007 at our center, and one of the main advantages of gallium-68 is that the DOTA chelator that is used can also be used for Y90 or lutetium-177, making this a true theranostic chelator, and so gallium-68 was actually the first theranostic PET radionuclide in clinical use. Over the past decades, many studies have shown that gallium-68 PET is clearly superior to indium 111 based scintigraphy. I invite you to look at the right side of this column, which lists up publications in which patients underwent both imaging procedures. On the right column, you see the difference in sensitivity, which ranges between plus 24% for gallium-68 to plus 62%. And so I think with this, there's no debate that one should be using PET, gallium-68 based, or with novel agents for imaging the somatostatin receptor in clinical patients. So making the use of the old Octreoscan scan labeled with indium 111 obsolete. And so you see that based on this difference in sensitivity, you will roughly detect the double amount of lesions as compared to the scintigraphy. If we now look at gallium-68, it has a number of advantages. You don't need a cyclotron. It's available from a germanium-68, gallium-68 generator with a half-life for the germanium part of 271 days. You can use chelation chemistry, which is compatible with biomolecules. There are kits that have been developed to use it, for instance, SOMAKit TOC. On the downside, you have a high positron energy of almost 1,900 megaelectron volts, which results in a large positron range up to 9.5 millimeters. There's a low activity yield, so you can only scan between 1 to 4 patients per PET camera, and you have fluctuating patients per production depending on the half-life of your generator, which can bring you from anywhere from 4 to 2 or even 1 patient per batch. And so it makes it very difficult to distribute gallium-68 labeled radiopharmaceuticals from one center to another one. And there's also a high fixed cost for the gallium generator, which ranges between 60 to 120,000 euros. And so there's a kind of gallium generator paradox. You do not need a cyclotron, but it does require radiopharmacists, and so often the gallium generator are still found in the cyclotron centers. Fluorine-18 can truly be considered as one of the ideal isotopes for PET imaging. The half-life is 109 minutes. It's ideal for imaging 1 to 3 hours post-injection, and it's nicely suited to the kinetics of many small molecules. The DK has a very high positron emission yield of 97%. Positron energy is much lower than that of gallium-68, resulting in a shorter range of 0.8 millimeters, so sub-centimetric positron range. There are no gamma emissions. The injected amount per patient is typically in the 100s of megabecquerels on classical time-of-flight PET cameras, and the absorbed dose is typically within 20 to 25 microsieverts per megabec. And very importantly, you can produce fluorine-18 in Curie amounts or in tens of gigabecquerels, and combined with the half-life, this allows for distribution, and one can scan into tens of patients per production batch. Disadvantage of fluorine 18 have to do with the harsh chemical conditions that are necessary for labeling. You need organic solvents, high temperatures, and sometimes time-consuming multi-step processes. And in particular, if you want to label complex peptides or heat-sensitive biomolecules, this can generate problems. And another disadvantage is that the fluorine radionuclide cannot be substituted by a therapeutic radiometal, so it has limited theranostic potential in theory. Big breakthrough was this chemical innovation proposed by McBride et al. In 2009, where fluoride will be reacted with aluminium 3+, and so that aluminum fluoride bonds, which are highly stable in vivo, are formed. And this aluminum fluoride complex can then be chelated in a second step by a chelator. And so this actually transforms the halogen fluorine in a pseudo radiometal, and which has the properties of this. So a proof of principle of this strategy was done with F18 labeled aluminum fluoride octreotide, and so this we see here the reaction scheme that was used, and this aluminum fluoride nota octreotide was then tested in biodistribution studies, where we see a biodistribution study with high tumor uptake that could be blocked by injecting cold octreotide, and this the uptake was similar to the gallium-68 labeled counterparts. There was low uptake in bone, which is an organ of interest, because if you have defluorination, free fluoride will of course go to the bone, and so you see here high tumor uptake comparable to gallium-68, and you see here an image in the mouse with a F18 ligand. This can be blocked by injecting cold octreotide, so showing that it's ligand specific, and then this is the same mouse where the tumor has grown, and that has then been imaged with a gallium-68 counterpart. So this seems to be working in mice, and it has the advantage, as already mentioned, compared to gallium-68, that with F18 you can have a centralized production and ship this to satellites, PET imaging sites, wherein the patient will be injected and imaged. So we started to take this very nice concept further and to try to get this into patients with an aim of validating it for clinical use. And so the first step that we did was to implement the GMP compliant synthesis of Alfoc at our center, and this has been done by our colleagues from radiopharmacy. And of course, once we had the tracer, we started doing biodistribution studies in healthy volunteers, and we did this in 6 healthy volunteers where we took sequential images. We see here images going from 4 minutes to 71, and we see the typical biodistribution of a somatostatin receptor ligand with uptake in the pituitary gland, in the spleen, and in the adrenal glands. We quantified this, and you see that the spleen is an organ with the highest uptake, similar to gallium-68 counterparts. We see high uptake in the pituitary gland, and we see limited uptake in the red bone marrow, which is a good sign, so that we also in humans we don't have signs of defluorination of this tracer. We computed the effective dose, and this is 22.4 microsieverts per megabecquerel, and which is very similar to the value of gallium dotatate, which is 21. So we are in the same range for injected megabecquerel. We then injected this to a first patient that came for a routine gallium dotatate scan that we see now on the screen. And these are the images of our very first patients injected in our trial with aluminum fluoride nota octreotide. And you see that we have a very similar images showing the liver metastases and showing some of the bone metastases with high uptake and high contrast. So this was very encouraging. We then injected another 5 patients. So in total, we scanned 6 patients. We compared with gallium dotatate images, and patients were scanned with the F18 molecule at 60, 120, and 180 minutes post-injection. And you see here that the uptake is rather similar in the different organs with F18 at the 3 different time points, and this is a little bit lower than that we see with the gallium-68 dotatate. So showing good uptake, and again you see that in the bone the values for F18 are actually lower than the values for gallium-68. Important to realize. We then looked at the detection of lesions in these metastatic neuroendocrine tumor patients, and we looked at the union of lesions, which were all lesions seen on both the gallium-68 scan and the 3-fluorine-18 scans, and we looked then at the fraction of those lesions that was seen on each individual scan. And I invite you to look at the 2-hour point for the F18 to compare this with the gallium-68. So you see that in 4 patients we detected more lesions on the F18 scan than compared to gallium-68. In 2 patients we saw somewhat more lesions on the gallium-68 than on the F18, and but overall there were more lesions at the 2-hour time point compared to the gallium-68, and this was an increase compared to the 1-hour time point for F18, and at 3 hours we did not see an increase in the number of lesions, so we go from 90 at 2 hours to 91.7, and so we decided that 2 hours was the ideal time point to study. If you look at the uptake in SUV values, you see that most of the points here cluster around the identity line. But there was one patient that yielded substantially lower uptake with the F18 ligand, and so this was something important that we took into account when we designed our multi-center trial. This work was awarded a Young Investigator Award at the ENM conference in 2021, and was also awarded the best paper published in the European Journal of Nuclear Medicine and Molecular Imaging in 2020. And we then went forwards to design a multi-center phase 2/3 trial to validate these findings in a larger patient group. This was done by PhD student Dr. Lynn Powells, and we actually used 2 arms in this trial. Arm A looked at PET-CT, and we had 75 patients in this arm. An interim analysis was planned after 20 patients with a backup plan in case of fertility, and arm B was using PET-MRI, and there the number of patients to be included was ranging between 10 to 20, and the trial started in October 2020. The knowledge at that time about ALFOC was based on our 6 patients that we had, and there was also a Chinese publication that had studied 22 NET patients, but there was no comparison with other gallium-68 ligands that was performed in this study. It was only compared to FDG. So the primary objective of this trial was to demonstrate the non-inferiority of ALFOC compared to gallium-68 DOTA somatostatin analogs. Main inclusion criteria were histologically confirmed NET of all grades were available, but we will see that there were very little grade 3 included. There had to be a less than 3 months interval with a routine clinical gallium-68 somatostatin receptor PET that could be either done before or after the experimental scan, and the patient had to have at least 1 known tumoral lesion outside of the head region. I'll explain later why the head region, with at least one of the following: so a size on morphological imaging of at least 1 centimeter, or an SUV max of at least 10 on gallium-68 DOTA PET. Secondary objective were to look at the detection rate at the organ level, were to look at semi-quantitative results looking at SUV and tumor to background ratio, to evaluate the clinical impact, to compare the F18 versus the specific DOTA somatostatin analog used to look at the influence of tumor grade, to perform an inter-observer agreement analysis, and finally to look at whole body MRI correlates, and to look at safety. And I will show you selected results of this trial. Primary endpoint, this was a non-inferiority trial, and so the primary objective would be met if the lower margin of the 95% one-sided confidence interval for the mean differential detection ratio is higher than minus 50%. So this was actually based on the assumption that ALFOC would yield a similar detection rate as the gallium-68 DOTA ligands. The minus 15% cutoff rate was determined in consensus by the study team, which included nuclear medicine physicians and clinicians, and we performed a power calculation with a 5% significance level that would show that if we include 75 patients, we would have more than 99% power if the true differential detection rate was 0%, 99% power if it was minus 5%, and if the true differential detection rate would be minus 10%, we would have 81% power to demonstrate non-inferiority. And so for arm A and B, the study procedures that were followed for the PET scan, we injected 4 megabecs per kilogram of Alfoc. Imaging was at 2 hours post-injection, and this was done for 3 minutes per bed position on our time of flight PET-CT and on 4 minutes per bed position in our non-time of flight PET-CT that we have at our center. The clinical routine scan was done, of course, with gallium-68. It was done according to the ENM procedure guidelines, and we injected between 114 on average and 139 megabecquerels per patient for DOTA-NOC and DOTA-TATE respectively, and patients were scanned at 2.5 minutes per bed position. So somewhat more activity was used for F18, but we do have more activity. That's one of the advantages of F18, and we scanned for somewhat longer than was done in the routine clinical setting. For the image analysis, the images were decapitated because we observed that there was substantially lower uptake of the F18 tracer in the salivary glands compared to DOTA-TATE, and so we did everything that we could to make sure that the readers were blind for the tracer that was being used. Also, the date and amount of radionuclide and the weight of the patient were all set to standard values so that this did not give away which tracer was being studied. So we had performed a truly randomized and blinded consensus read with 2 observers to determine the amount of lesions per patient. Very importantly, the differential detection ratio, I already mentioned it during the primary endpoint. What is this? Well, this is simply the amount of lesions that are detected by the F18 tracer, of which we subtract the amount of lesions detected by the gallium-68 tracer, so which is a detection ratio. And so importantly, a higher value shows a benefit for F18, a value below 0 shows a benefit for gallium-68. So these are the results of arm A, the PET-CT arm. So in total, 75 patients were recruited. Median age was 65 years. 61% were men. Primary tumor was mainly intestine, 60%, and pancreas for 24%, and then some other tumor lesion sites. The tumor grade was grade 1 in 35 patients, grade 2 in 34 patients, and there were only 2 patients that had grade 3. So our results, of course, are valid for grade 1 and grade 2, but not for grade 3 patients. 3/4 of patients were scanned with gallium DOTA-TATE, and 1/4 or 19 patients was scanned with DOTA-NOC as a routine clinical tracer. And importantly, the interval between both scans was a median of 7 days. You see here the distribution, and this ranges from minus 30 days to plus 32 days. So some patients, the F18 scan was done before the clinical scan. The primary endpoint, as already mentioned, so for this, the 95% confidence interval lower bound should be higher than minus 15%. And so this is the main results of arm A. We detected in total, in a blinded read, 4,278 lesions with the F18 molecule and only 3,454 lesions with the gallium-68. In total, if you take the entire union of all lesions, 4,709 lesions were detected. And so this results in a detection ratio of F18 91.1% of those 4,709 and 9 lesions in the union, and with gallium-68 only 75.3% of these lesions were detected. So this results from 9.6% to 22.0%, and so this meets, of course, the primary endpoint, and this is also showing actually superiority. We see an example of a patient in arm A. We see lymph nodes in the chest. We see large lymph nodes in the abdomen, but we see also that there are some peritoneal metastases, which are clearly more better depicted by the F-18 images than by the gallium-68 images. This is another patient where we observed an incremental lesion in a rib. A patient also has liver metastases and lymph node metastases in the abdomen, but this rib metastasis was only shown on the F-18 scan. And if you look at the scan on the right, there you see a DOTANOX scan that was done 6 months after the previous DOTANOX scan, the one that was used for the trial. And there you see that this lesion now is also visible on the DOTANOX scan, which confirms that it is a true positive incremental finding on the F-18 scan. If you then look at the secondary endpoints, if we first look at the site where lesions have been detected, of course, for all sites there was an advantage. This advantage was also seen for liver, for detection of lymph node metastases, lung lesions, peritoneal lesions. We observed non-inferiority for pancreatic lesions, but without demonstrating superiority at that site. And finally, bone metastases, we could not demonstrate superiority nor demonstrate non-inferiority, but of course the trial was not powered to demonstrate this. If you then look at the different gallium-68 tracer that has been compared with, you see that for both DOTATATE separately and DOTANOX separately, you see that there is a positive differential detection ratio, 11.8 and 27.5 respectively, showing again superiority for F-18 tracer. And also if you look at grade 1 or grade 2 separately, you see that again, we have both non-inferiority and superiority, which is demonstrated. So we think that regardless of the subgroup that we look at, the results observed in the entire group are still valid. If you then look at the semi-quantitative analysis, you see that the SUV max was slightly higher in the gallium-68 group, 22.4 versus 20.0. This was not statistically significant, as we can see. P was 0.067. But if you look at the tumor to background ratio, you see that the F-18 has a higher tumor to background ratio, 31.7, compared to gallium-68 at 25.1, which of course is one of the drivers of the higher lesion detection ratio, and this was statistically significant. If we then look at SUV max analysis, you see that there's a good correlation between both the SUV max and both the tumor to background ratio between the gallium-68 and the F-18 tracer. And so in summary, we can say that the mean differential detection ratio was positive in favor of F-18 and with a 95% confidence interval above the pre-specified threshold of -15%. So aluminum fluoride node octreotide is non-inferior and even superior compared to gallium-68 DOTATATE and DOTANOX. We consider this to be a validated alternative for clinical practice somatostatin receptor PET, and regardless of the lesion site, except for bone, regardless of the grade 1 or grade 2, and regardless of the comparator you want to make, DOTATATE or DOTANOX, these results are validated by the subgroup analysis. And this work at the latest ANM almost 1 year ago received the Marie Sklodowska-Curie Award 2022 for best research abstract submitted to the Congress. If we then look at the results of our arm B, there we used a different imaging modality. We used PET MRI. And why did we did this? Well, we wanted to be able to look at a morphological correlate of the lesions that were detected by the F-18 tracer. And we actually wanted to use the signal from the MR to confirm that the lesions that we pick up with the F-18, and in particular the incremental lesions, are true neuroendocrine tumor lesions. So in this arm, we included in total 10 patients. The median time between the two scans was 13.5 days, so somewhat longer than for the arm A, and it were only grade 1 and grade 2 tumor grades. You see here the results of the lesion detection. We see that the mean detection ratio for gallium-68 was 91.4 if we compare to the union of F18 and gallium-68. For F18 this was 99.1, so the differential detection ratio is 7.7. We see the 95% confidence interval, it goes from -0.4 to 15.8, and so the p-value for non-inferiority was highly significant, so this is non-inferior. The p-value for superiority was not significant. In total, 195 different lesions were detected, 193 with the F18, 167 with gallium dotatate, and so this was non-inferior, but we could not prove superiority. Also, this trial was not powered to do this, of course. But more importantly, if we look at the MR correlates, we see that of the 193 lesions detected on F18, 185 had a corresponding lesion on MRI. This is 95.8%. 4 only had physiological uptake, and 4 did not show an MR correlate. So we keep 105 lesions with an MR correlate, and 178 of those were net lesions. This is 96.2%. So we call these MRI confirmed net lesions, and there were 7 benign lesions that were not deemed to be net lesions, 3 myofibromas in the uterus, 3 schwannomas, and 1 hemangioma. This is an example of a patient scanned in arm B. You see here on the left F18 images. We see 4 lesions that are depicted on the PET, and that also have a correlate on the diffusion weighted MR. And then you see here the gallium dotatate images, and we see the same 4 lesions. So this is a patient where we have concordant findings. This is a patient where we have incremental findings that we see on the right. You see that the lesions depicted by the blue arrow on the left, which is the F18, are also seen on the right by the gallium-68 tracer, but the lesions that are depicted with green arrows in the F18 image are not seen on the gallium-68 dotatate lesions, and this is what we call incremental lesions. In total, we observed in this cohort of 10 patients 36 incremental lesions. 33 were seen on the F18 scan, 3 were seen on the gallium dotatate. This was mainly liver and bone lesions, and if you then look at the MRI correlate of the 33 incremental lesions that were detected by the F18 tracer, you see that 30 of those were actually confirmed net lesions. 2 had no MRI correlate and 1 was a schwannoma, but more than 90% of the incremental lesions that we see with the F18 tracer are confirmed by the MRI. And that was the main aim of this trial, and this is another example. You see on the upper left an F18 image. There are 2 lesions that are seen in the liver. If you look at the image below on gallium dotatate, you see that only 1 is seen. That's the one marked with the blue arrow. The one with the green arrow is not seen on the gallium-68 image. If you look at the diffusion weighted image on the upper right, you see that both lesions are seen, the green and the blue arrow detected lesion. And on the lesion at the lower right position, you see the DOTA-NOC that has been repeated 6 months after the dotatate scan that was used in the trial. And there you see that the incremental lesion first picked up by F18 is now also seen 6 months later by the gallium-68 radiopharmaceutical. So in conclusion, for arm B, we showed that F18 ligand was non-inferior to the gallium dotatate. Superiority was not confirmed because the trial was not powered to do so. We only had 10 patients, but more importantly, more than 92% of the F18 detected lesions were MR confirmed net lesions. And if you then only look at the incremental lesions, the ones that were not seen by gallium-68, more than 95% of those were confirmed by MR. And we think that this further validates F18 aluminum fluoride octreotide as an option for clinical practice SSTR PET. If you now look at our future work with this tracer and our conclusions, Future work will be some further exploration of secondary endpoints. This will be done by Hannes Leupe. We will look at the clinical impact of the F18 tracer compared to gallium-68, and for this we've organized a number of tumor boards where patients are presented and information of both image tracers and images are provided. We have analyzed 75 out of 75 cases, and we will present the results of our first 54 analyses at the upcoming EANM 2023. And then we are currently performing readouts for an inter-observer reproducibility study. You see here the name of the people that are doing the readouts, and we hope to present this in the course of 2024. Importantly, we submitted a dossier to obtain approval for clinical use of this tracer to the Belgian authorities, and we have received this in March of this year based on the results of Arm A of our trial. And based on this, we can now work on the submission of a reimbursement dossier in Belgium, and we hope to do this by September or October. And if things go well, 6 months later, we will have full reimbursement of this in Belgium. This work is generated quite some attention in the literature in F18 labeled somatostatin analogs. There have been invited comments, both in the European and by colleague Motegi, who calls it the battle of David versus Goliath, where F18 would be a challenger of the giant gallium-68 Goliath. But I wonder, if you look at the ubiquitous use of F18 in nuclear medicine, if the true giant is not the F18 in this setting. There have been comments that ALF18F is phagocyting the clinical gallium-68 applications, and also recently our radiology colleagues have picked up our work and stating that the F18 can improve the staging. We've tried to summarize the current state of the art of the literature in this recently published review in JNM, which I invite you all to read. And importantly, in that review, we also look at other molecules. For instance, cifadendate has also quite some substantial literature supporting its use. So it's not just aluminum fluoride notooctreotide, but given the nature of this webinar, I focused on this tracer. And to conclude, fluorine 18 plays a historically very important role in PET imaging. Of course, it's the radionuclide for FDG. The physical characteristics and logistics for PET are optimized for fluorine 18. Classical or novel radio labeling strategies using alpha 18 allow to provide facile labeling of biomolecules, and so can now used as an alternative to gallium-68 and used in diagnostic duo. And specifically for alpha notooctreotides, we see a similar biodistribution and dosimetry as for gallium-68 somatostatin analogs. We detect more lesions in our prospective multicenter trial with a centralized blinded readout. We observe higher tumor to background ratio independent of organ site, WH grade, or the DOTA SSA to which we compare. We validated these findings by using diffusion weighted MRI, and more than 95% of our lesions are confirmed as being net, and so we consider this to be validated for clinical practice based on these results. It is approved for use in clinical patients in Belgium, and reimbursement is still something that we are currently working on. I would like to thank the following collaborators, and in particular Kom op tegen Kanker, Belgium charity that has actually funded this entire line of work that I presented, and I will be happy to take questions at the end of the seminar. Thank you very much for your attention.
Thank you very much, Christoph, for this excellent talk and this really impressive results. I think we have seen that alpha-notooctreotide is really not inferior and even superior to gallium dotatate, and this work also laid the base for our project here in Chile and the possibility to implement this tracer here in our country. I know it's late, so hopefully you can hold on a little bit longer. I'm sure we have some questions for you at the end of the talk. I have one comment to all those connected from home. Please ask your questions in the chat. We'll collect them and then ask them later on. para todos que están conectados desde la casa o desde el trabajo de otro lado, por favor escriben sus preguntas en el chat, nosotros los colectamos y los vamos a hacer al final del webinar. Bueno, eso dicho, vamos a la siguiente presentación. Hemos visto los datos clínicos, pero todo eso también requiere que nosotros podemos producir estos radiofármacos en actividades, en cantidades suficientes para la distribución a larga distancia y en suficiente cantidad en Chile. Entonces, nosotros 2 o 3 años atrás empezamos con este proyecto sintetizando bajas cantidades y en conjunto con una colaboración con IVA en Bélgica, nosotros logramos de escalar esta síntesis, de mejorar diferentes parámetros y de eso les va a contar nuestra próxima presentadora. Noemi Emanuel es radioquímica, trabaja en IVA, en Louvain-la-Neuve, Bélgica, encargada de investigación y desarrollo y nos va a presentar sobre ALF Chemistry on Centera Plus and Automated Access for ALF Peptides. Noemi, please.
Hello everyone, I am Noemi Emmanuel. I'm radiochemist for IBA, and I would like to start by thanking Dr. Vasco Kramer for the invitation so that I am able today to talk to you about aluminum fluoride chemistry, how this can be a really helpful tool, and how you can implement it in your facility with the Centera Plus. So a small disclaimer. First, I am a full-time IBA employee, which means that even if this presentation is going to be more scientific, I will still talk about the IBA equipment. But that being said, let's dive further into the aluminum fluoride chemistry. Why is it that interesting and how this can be helpful for you and your productions? So we all already heard the debate between isotopes, fluoride on one side, radiometals on the other side, which one is the best isotope for which application. They both have very good features. 18F has a very convenient half-life for production, and it has a high percentage of low energy positron, which help to obtain a very good resolution on the imaging. 18F is usually bonded to the tracer by a covalent bond by nucleophilic substitution, which requires to heat and takes a little bit of time. On the opposite, radiometals doesn't need to be covalently bonded, but is bonded through chelation. Chelation is a little bit faster and can be performed at room temperature. So you don't necessarily need to heat it, which means that it can be good if you have sensitive biomolecules, for example. Nowadays, there is a variety of efficient chelating groups, so you have a choice between all of these. And once your protocol is established for one chelating agent, it's really easy to adapt the synthesis to other peptides. You just need to adapt a little bit the process. But aluminum fluoride chemistry is really a good solution to combine the strength of both these isotopes, as aluminum fluoride is considered as a pseudo-radiometal, so you will have both the advantages of the radiometals with the easily adaptable protocol because of the chelating agent, but you will keep a very good quality of imaging because of the fluoride. Now, the fluoride is bonded to aluminum quite strongly. Actually, this is the strongest bond amongst all other halogens. So the challenge here is most to select the right chelating agent. In the literature, we usually see NOTA as a good chelating agent, and recently there have been a few studies also on NODA, which is a derivative of NOTA, and it gave excellent results, so this is also a possibility for your chelation. But the critical point is really to work with a good quality of fluoride. So a fluoride that will contain the less metals as possible, as those metals could be in competition with your aluminum, and you don't want this for your chelation. You will have a really bad yield if you have other metals taking the place of your aluminum. So that's really something to be careful about. The first aluminum fluoride protocol was described by Laverman and McBride in 2010. And since then, it has been modified, it has been reworked by other groups, but the skeleton of the process remains the same. So it starts with an incubation time. The incubation is the time necessary to form the aluminum fluoride complex. So in this step, you have the aluminum source, the aluminum chloride, that is mixed with aqueous fluoride, and it formed the complex. Only after this, you will add the peptide with the chelator contained in a buffer for the labeling, a buffer, for example, acetate buffer, because here you really need to control the pH accurately, I would say. Because if you are too low, you will create HF, and if you are too high, you will create hydroxide of aluminum, and this is really not desirable. So you heat the solution for 10 to 15 minutes, but the real advantage here in terms of time is that you do not need to perform the azeotropic drying. You can use the fluoride without it being dried, and this is really an advantage of the aluminum fluoride technique, that it's really tolerant to water content. After the labeling, you wait a little bit for the solution to cool down, and then you dilute it to trap it on a cartridge for SPE purification. You elute the cartridge with ethanol, and then you obtain your product that you can put in any final formulation you desire. Usually, people use a buffer to keep a good pH. So now, how can you implement this technique for your production in your facility? So IBA developed the Stintera Plus, which is an automated module that is used for the production of a diversity of radio tracer. It has a really small size, which allows it to fit in any type of hot cell. The electronic of the modules are outside of the hot cell, which protect them from the radiations and help the module to live longer. And you yourself, you can control the module with the software with a safe distance between you. You don't need to interact with the module immediately. The synthesis will be performed in a single-use plastic cassette that we call an IFP. So every step of your synthesis will be performed inside of this plastic cassette, which is really convenient for aluminum fluoride chemistry. You can also use it for R&D purpose. If you want just to modify it, it's really simple. It's just a bunch of tubing plugged inside the valve, so you just need to take them out and plug them differently. It's really easy for you, but you can also just use it for production. It will respect the GMP requirements. So we use this equipment to adapt the classical process of aluminum fluoride to fit into the Stintera Plus. So we decided to choose a one-pot labeling, which means that we do not wait first 5 minutes for the incubation and then 10 minutes for the labeling, but we rather mix everything together, the precursor, the aluminum, and the fluoride all together for 5 to 10 minutes, and it's really efficient. We also could skip the cooling to room temperature because the volume we use for the dilution is quite high and it reduces the temperature already significantly. So you do not need to wait for the cooling, it saves time, and you lose less product by decay. So this is what the IFP would look like. Please note that this is not commercial as yet. It's still under development. And what we would like to do is to have this same IFP for any type of peptide that would be done by aluminum fluoride chemistry. For now, we tested it with FAPI74 and notautriotide during our collaboration with Sophie and PositronPharma, respectively. Again, a small disclaimer. As I said, we want it to be available for any type of peptides, but be careful that some peptides are protected by patents, and so make sure you have the authorization to produce the peptide you desire to do on your site. That being said, if you take a look on the right in the table, it describes the different components of the IFP. You have first the QMA element, then you have the precursor solution that contains the precursor, the aluminum source, and the buffer. Then you have the HLB element, the buffer to clean the cartridge, both cartridges, the reactor, and on the far right of the IFP, you have the QMA cleaning solution, which is just ultra pure water that is used to clean very thoroughly the QMA to remove as much metals as possible to avoid competition with the aluminum. During our research, we discovered that having a labeling at 100 Celsius degrees was much faster than at 70 degrees, which makes sense, so we decided to keep that condition. And regarding of the time, we tested from 5 minutes to 20 minutes. And as the difference was not really significant between those times, we decided just to keep the shortest time possible. For the dilution, we also noticed that the volume used for it was really important, as the content of organic solvent that you have in your vial could be a problem for the trapping afterwards on the HLB cartridge. So you really need to dilute with a significant volume of buffer before trapping. So with this configuration, we could obtain a yield around 30 to 60%, depending on the starting activity. For the QC, we decided to follow the guidelines for FDG, as there is no pharmacopoeia monograph for either of these tracer, FAPI or notautriotide. And we had no problem with that. You see the chromatograms for the reference product and the UV where we detect the ascorbic acid. So we had everything was compliant for us. It was the same for GC. Only classic solvents to be detected: methanol, ethanol, and acetonitrile. Please note that in this table, the values are given before dilution with the final formulation, so they are just a bit higher than what it should be. Radiochemical purity was also good, above 98%, so no problem with that. So all of these were results from our research lab. But now I would like to show you that this technique can be and is implemented in real production in PositronPharma in Chile and R2IBF in Brazil, where they produce aluminum fluoride notautriotide for the diagnosis of neuroendocrine tumors. So gallium dotatate is well established for the detection of neuroendocrine tumor cells, as it has a high affinity for somatostatin receptors. The problem is that because of its short half-life, it's very difficult to distribute this tracer in countries such as Latin American countries, which are often very vast, big, or very long. You have to cross long distance to distribute your tracer. So that's why R2IBF and PositronPharma decided to turn to aluminum fluoride notautriotide because it showed comparable efficiency for the diagnosis of the NET tumor, but also has the excellent image quality of the fluoride. Today, there are at almost 100 production of notautriotide with the Sintera Plus, and they distributed more than 1,000 doses to Latin American people, so it's really impressive. These results were very recently published in a cancer's journal, so if you didn't take a look already, I recommend you to do so. So you may recognize on the right the IFP we were talking about before. So you can see the detail of the vial for the QMA cleaning to reduce the amount of metal present in the fluoride. They also use the one-pot technique, so the aluminum chloride is already mixed with the precursor in the vial, so before the reaction. And here they also add a little bit of ascorbic acid, which is a great idea to reduce potential metals that could still be present during the labeling. So the reaction was heated at 100 Celsius degrees here for 10 minutes, and then purified via the cartridge SPE purification. So on a total of the 99 production that were performed during 10 months, or just 10 months, they just needed to discard it for production that were out of specification, but for the rest it was compliant, and they obtained an average yield of 31%, starting from an average activity of 165 gigabecquerel, which results in the average production of 52 gigabecquerel of aluminum fluoride notautriotide. And all these were distributed to more than 1,000 patients over 10 different PET centers and on a distance of more than 1,000 kilometers of distance, which is quite amazing and would probably be very difficult to obtain with gallium and its short half-life. So it's really due to the half-life of the fluoride that you can do such a thing. For the QC, again, because there is no monography for notautriotide, they decided to respect the dotatate criteria. So you can see here the value for the remaining solvents, and on the right, the chromatograms from HPLC. Here, because of the condition of injection, they could see separatively the two stereoisomers of notautriotide, and the final product was stable for more than 8 hours, which is very convenient for distribution. It has to be stable to travel long distances. And here is the detailed table of the criteria they respected to determine if the batch was compliant or not. And with these criteria, on the 99 batch produced, 95 could be successfully injected to patients. So I hope that with this presentation, I could convince you that aluminum fluoride chemistry is really an interesting tool, and pseudoradiometals is something that can change the way we produce radio tracers. That in that matter, Sintera Plus can help you for your research on your production of aluminum fluoride peptides. But most of all, I hope you saw how the production of aluminum fluoride notautriotide could really open the access to Latin American people that were further from the production center, and it gives them the access to diagnosis, and it's really a nice thing to have for anyone in the world. So if you have any specific question, you can contact me. And again, I would like to thank Vasco Kramer for the invitation, and thank you all for your attention. Thank you.
Well, thank you very much, Noemi, for the great presentation and also for the great collaboration during the last years. This was really helpful for us to implement this new tracer, and thank you for that. As well, hold on, we are done in 15 minutes, and then we have a couple of questions, and I guess also for you. Todos los conectados en casa, por favor, envíen sus comentarios, preguntas al chat y nosotros los colectamos y hacemos después. Bueno, ahora después de que hemos visto el trabajo que hicimos en conjunto con IVA, nosotros teníamos ese nuevo trazador disponible para estudios clínicos en Chile, en pacientes, y lo primero que hicimos era un estudio colaborativo con PositronMed, una translación clínica, por decir, aquí en Chile, creando evidencia local, un estudio comparativo que nos va a presentar doctora Arlette Heger. Arlette Heger es especialista en medicina nuclear, trabaja hace 8 años en PositronMed. Recientemente hizo un fellow de investigación sobre neuroimágenes en la Universidad McGill en Montreal, California. Y Arlette, bienvenido y adelante, tu presentación.
Ok, muchas gracias, Vasco. Bueno, voy a hacer la presentación en español, por si acaso. Buenas tardes a todos. Primero voy a hacer un breve resumen del trabajo que publicamos a principios de año y también presentamos en el Congreso Europeo de Medicina Nuclear, para después mostrarles imágenes de pacientes que incluimos en este estudio y así puedan hacerse una idea de cómo se ve realmente y comparativamente el galio dotatate versus el fluor octreotide. Primero nosotros nos planteamos como objetivos principales evaluar la biodistribución del fluor octreotide versus el galio dotatate. Quisimos comparar la captación de las lesiones neuroendocrinas y esto lo hicimos a través de la medición de los SV máximos con ambos radiotrazadores. Quisimos evaluar cómo se comportaban en la relación lesión-tejido de fondo y esto lo hicimos con hígado y vaso. Y cómo se comportaban estos trazadores en la detección en el número de lesiones que presentaban los pacientes. Para eso incluimos 20 pacientes con una edad media de 57 años, todos tumores neuroendocrinos en etapa 4 que fueron comprobados por biopsia y adquirimos imágenes PET-CT de galio dotatate y fluor octreotide. Posterior a la inyección, se tomaron las imágenes de alrededor de 60 minutos posterior a la inyección y fueron desde cabeza hasta la raíz de los muslos aproximadamente. Y lo hicimos en nuestro equipo Siemens Biograph Vision. El intervalo de tiempo entre ambas imágenes, entre ambos PET, fue de una media de 12 días, que va con un rango de entre 2 a 30 días. Para eso hicimos los VOI, los volúmenes de interés, en las lesiones que eran consideradas tumorales y las distinguimos visualmente como regiones que captaban mayor el trazador en relación a la captación de fondo o la captación fisiológica que se espera para el radiotrazador. 2 médicos nucleares que obviamente no estábamos cegados a los datos clínicos, analizamos de forma independiente las imágenes. Comparamos los perfiles de biodistribución de ambos trazadores en los órganos normales y esto lo hicimos con la medición de SV min y SV max. Y para evaluar la captación en las lesiones propiamente tales lo hicimos a través de SV max y SV peak. Y también evaluamos la relación de lesión tejido de fondo, ya sea con el hígado o con el vaso, y eso lo hicimos dividiendo los SV max de las lesiones por el SV min del hígado y del vaso respectivamente. Entonces, primero, para evaluar la biodistribución del fluor octreotide, comparamos la captación fisiológica en órganos normales y basados en el SV min de estos tejidos y encontramos que, al igual que el fluor octreotide, tenemos mayor captación en el hígado, en el vaso, en las glándulas suprarrenales, en el riñón, pero esta captación es significativamente menor en algunos casos en comparación a la captación de fondo o de estos órganos con el galio dotatate. Uno de los hallazgos también que más nos llamó la atención es la menor intensidad de captación a nivel intestinal, que después vamos a ver en las imágenes, que es significativamente menor con el fluor octreotide versus el galio dotatate. la otra área que evaluamos fue cómo se comportaban las lesiones hipercaptantes en relación a la captación de fondo, ¿ya? Y esto lo hicimos con el hígado y el vaso. Aquí agrupamos las lesiones neuroendocrinas en el tumor primario, en el tumor primario, las metástasis hepáticas, óseas, adenopatías y otros sitios. Otros sitios incluimos ahí carcinomatosis, metástasis pulmonares y metástasis en otros sitios. En casos en que los pacientes presentaban múltiples lesiones o lesiones innumerables, consideramos al menos 5 lesiones por grupo de metástasis para evaluar un promedio de SV máximo, ¿ya? Y la verdad es que no encontramos diferencias significativas entre ambos radiotrazadores, si bien las lesiones tienden a tener un SV máximo menor con el octreotide versus el galio dotatate. La captación de fondo en el hígado y en el vaso también es menor, por lo tanto la relación no es diferente entre ambos radiofármacos. Y por último, para evaluar las lesiones detectadas con ambos radiotrazadores, nos dimos el trabajo de ir contando las lesiones de cada paciente. Obviamente las lesiones innumerables no las podíamos contar. Sí encontramos que en 4 pacientes habían entre 1 o 2 lesiones que no captaban con fluoroctotide y sí captaban con galio, pero esto en ningún caso significó un cambio de etapa del paciente o un cambio de pronóstico del paciente. Y en un paciente sí vimos una lesión hepática específicamente que captaba definitivamente con el octreotide y que no era visible en el galio dotatate, por más que ajustáramos la ventana o el ruido propio de la captación hepática con el galio dotatate, no fue posible encontrarla. Entonces, a continuación les voy a mostrar algunos pacientes que evaluamos en este estudio para que se hagan ustedes su propia idea de cómo es la distribución del fluoroctotide versus el galio dotatate, para que vean cómo captan las lesiones primarias, las lesiones metastásicas y creo que solo recuerden que estos pacientes se hicieron con una diferencia de menos de 30 días entre ambos PET y no recibieron ningún tratamiento entre medio de ambas imágenes. En las imágenes superiores vamos a encontrar el PET con fluoroctotide y en las inferiores el PET con galio dotatate. Esta primera imagen corresponde al paciente número 3 de nuestro estudio. Es un hombre de 49 años con un tumor neuroendocrino de páncreas metastásico que tenía un KI67 de 8%. Ya en el MIP pueden darse cuenta que en las imágenes superiores tenemos el fluoroctotide, se ve claramente porque existe casi nula o escasa captación en glándulas salivales. La captación de fondo del parénquima hepático, si ustedes la ven, en comparación al galio dotatate también es inferior. Esto lo pueden ver en las imágenes en el MIP. Pero en ambos PET, en ambas imágenes, podemos identificar claramente las lesiones hepáticas con ambos trazadores, ¿ya? Y este es el tumor primario del paciente, es un tumor de cuerpo y cola de páncreas que capta intensamente con ambos trazadores y también tiene un ejemplo de una lesión hepática intensamente captante igual con ambos trazadores. La segunda imagen corresponde a nuestro paciente número 4, es un hombre de 44 años con un tumor neuroendocrino de páncreas. Arriba tenemos las imágenes con fluoroctotide, abajo con galio dotatate. Lo mismo, la captación de fondo en el parénquima hepático es bastante menor con el fluoroctotide versus el galio dotatate. Sin embargo, igual se ven las lesiones hepáticas bien definidas. Y acá en esta imagen solamente quería mostrarles cómo es la intensidad de captación con ambos trazadores. Este es el tumor primario, es un tumor de cola de páncreas que ambos trazadores captan intensamente. Y además este paciente tenía una pequeña adenopatía retroperitoneal que está en este sector y ambos trazadores también la ubican perfectamente. Este es nuestro paciente número 12, es una mujer de 57 años con un tumor neuroendocrino de colon metastásico. El primario en este caso estaba resecado y como pueden ver en las imágenes MIP presenta lesiones secundarias hepáticas que se detectan con ambos trazadores y espero que logren ver también que el paciente también presenta implantes peritoneales hipercaptantes y lo que llama la atención acá, que quiero que se fijen, es la captación intestinal. Es bastante menor con el octreotide versus el galio-dotatate, lo que permite también en estos casos identificar pequeñas lesiones que van a ser hipercaptantes porque el fondo es mucho menor. En este paciente todas las lesiones hepáticas, quizás hay un desfase en el corte, pero todas las lesiones hepáticas que este paciente tenía captaban con ambos trazadores y eran plenamente identificables. Este es nuestro paciente número 13, una mujer de 60 años con un tumor neuroendocrino de intestino G1. También estaba resecado y si ustedes pueden ver presenta extenso compromiso hepático, pulmonar, adenopático, suprainfradiafragmático y también lesiones óseas secundarias. Solamente quería mostrarles que la captación de las lesiones hepáticas en este paciente, que eran innumerables, también este paciente no presentaba diferencias significativas en el número de lesiones identificadas. Nuestro caso número 11 es un hombre de 57 años, es un tumor neuroendocrino de primario desconocido y presenta innumerables lesiones hepáticas y también óseas secundarias. En este caso no ajustamos la ventana para ver las lesiones hepáticas, por eso ven ustedes acá que la captación es intensa en el parénquima hepático, pero solo quiero que vean la distribución de las lesiones óseas con ambos trazadores, que las detecta perfectamente sin diferencias en el octreotide versus el galio-dotatate. Y este es uno de nuestros casos controversiales que encontramos en nuestros 20 pacientes. Es el paciente número 2, es una mujer de 45 años que tiene un tumor neuroendocrino bronquial metastásico con un KI 67 de 1%. Los PET aquí se adquirieron con 7 días de diferencia, entonces este paciente no recibió ningún tratamiento entre medio, pero claramente ya en las imágenes MIP ustedes pueden ver que las lesiones hepáticas captan significativamente tienen menor intensidad en relación al galio-dotatate. Si bien el octreotide las logra detectar, la diferencia en la intensidad de captación no tiene una explicación, ¿ya? Y lo otro es que este paciente, si ustedes se fijan, tenía una pequeña, no tan pequeña, pero tenía una adenopatía subcarinal que en el galio-dotatate captaba intensamente y en el fluor octreotide captaba, pero también de menor intensidad. ¿Cuál es la explicación para estos hallazgos? La verdad que escapaba de este trabajo. No tenemos una confirmación histológica de estas lesiones específicas o de la adenopatía subcarinal, por lo tanto, no sabemos qué diferencias pueden tener estas lesiones en este paciente. Y el último caso es nuestro paciente número 10, es un hombre de 42 años con un tumor neuroendocrino hepático que tenía un KI 67 mayor al 20% y presentaba lesiones hepáticas, óseas, adenopatías suprainfradiafragmáticas, que las podemos ver muy claramente en las imágenes en el MIP. Pero si se fijan bien en las imágenes con fluor octreotide, hay un pequeño foco hepático que es claramente evidente, es hipercaptante, pero en el parénquima hepático con galio-dotatate no logramos identificarla. No tenemos una resonancia de este paciente para tener un mejor correlato anatómico y para ver si es realmente una lesión secundaria o es otro hallazgo. Y bueno, para concluir hay que destacar, como vieron en las imágenes, que las ventajas de tener un trazador marcado con flúor nos proporciona una excelente calidad de imagen. Vamos a tener con menos ruido en relación a la captación del galio. Tenemos una biodistribución similar al galio dotatate, con un poco de menos intensidad en la captación de fondo, pero visualiza las lesiones neuroendocrinas con alta sensibilidad. Y no se observó que el fluoroctotide sea inferior en comparación al galio dotatate en la detección de lesiones. Muchas gracias.
Bueno, muchas gracias Arlette y gracias a todos los presentadores. Ahora pasamos a nuestra sesión de preguntas, Q&A. And first let me check, Christoph, can you hear us? Greetings from Chile. Estamos esperando que tenemos conexión con nuestros invitados en Europa, en Bélgica. Ya tenemos la 1:15 de la noche. Okay, so we have to unmute Christoph. While we do that, Christoph, so since you can hear me, really, I want to extend our gratitude and thank you for supporting our webinar and for all the work you have done. So this has really been a milestone work, and I remember 2019 or 2020 when you published the image of the month in the European journal. I think this image gave us the kick and the motivation to start also to work on nota octreotide, and your work you have done has very helpful to implement this new product. So thank you very much for that.
Thank you very much for these kind words. I don't know if you can hear me now.
Now we can hear you. Perfect. So greetings to Leuven. And
yes, thank you very much. And I'm really pleased that the image of the month in the European also gave you a kickstart, because that's exactly what I think those images of the month should be, trying to open new technology and new avenues. And I'm really pleased that you scanned more than or around 1,000 patients with somatostatin receptor PET using the F18 molecule in Southern America, because I think this is really the main message that I want to convey, is that we should be scanning these patients with somatostatin receptor PET. I think that gallium-68, of course, dotatate, dotanok, are very good molecules that have been very important when we started with those in 2007 in Leuven. But the entire work that we did was to try to get alternatives for those patients where gallium-68 was not available, and to try to overcome the use of what at that time was still common, the indium-111 peptidreotide, so the octreoscan, which really will not give you the same image of your patients. And so that was the motivation to get this work started, and we were very pleased with the results that we got. So we tried to prove the equivalence of this tracer, and I think there's no doubt that we have proven this, but also, we see somewhat more lesions. We can debate about what the reason is there, why we see this. But I think there was a question in the chat about the theranostic use of the aluminum fluoride. I don't know if you want me to answer this question.
exactly, sure. Yeah. So the question, I think, was since aluminum fluoride nota octreotide seems to be superior to gallium dotatate, wouldn't it be a good approach to use lutetium nota octreotide as well for the better contrast and uptake?
Yes. So there, I think this has been also pointed out by the radiopharmacist talk, that you cannot use a DOTA chelator for the aluminum fluoride. So that's why McBride and Avaman, when they developed the technique, they switched to another chelator. And we actually used this molecule because that was the one that we could get the permits in Belgium to use in our patient work. So I think that part of the reason why we detect more lesions is that we have a lower liver background. I think this has also been shown in the South American experience. And so you can get some better contrast there, and you will see somewhat more lesions. But another reason is also in our protocol that we use, we inject somewhat more activity, so we injected 4 megabecs per kilo, whereas we injected clearly less with the gallium-68, also because we don't have that much gallium-68 when we produce a batch, and we need to scan at least between 2 and 4 patients per production because we have quite high demand. And also we scanned a little bit longer, and our time point was at 2 hours after injection, so we allowed some more clearance of the tracer. I think part of it is so not just, let's say, superiority of the radiopharmaceutical, but it's superiority of the entire imaging protocol that we have used. And so for the switching to lutetium, NOTA, octreotide, I don't think that we have a good case for that. Clearly, we haven't proven it for sure. And we simply use the F18 compound now as a theranostic partner with lutetium dotatate that we are still providing to our patients, for which, of course, we have all the data and we have all the permits and requirements. I like to point out in this setting that the NETTER-1 trial, where we also were investigators, their patient selection was done with theranostic imaging by using still the old school optional scan. All the patients needed to have an indium 111 pentatriotide scan. At the time we were doing gallium dotatate, and we needed to repeat the scan to satisfy the inclusion criteria of the NETTER-1. So NETTER-1 has proven the pair indium 111 lutetium 177 pentatriotide and dotatate. So I think using a positron emitter that is an agonist of the somatostatin receptor, not an antagonist, but it is an agonist that has shown clinical efficacy and has shown you as good images that you get as with the gallium-68, as I think we have now data for the F18 aluminum fluoride NOTA octreotide, but we also have this kind of data, for instance, for copper-64 dotatate, which I haven't mentioned.
It's also a nice one, yeah.
Yes, certainly. I think these are all molecules that I consider to be equivalent. Just as for the gallium, we consider DOTANOC, DOTATATE, and DOTATOC in clinical practice to be equivalent. So now we can add these new kits on the block. But for the therapeutic molecules, there clearly we stick with the lutetium dotatate, and there we have all the data from the NETTER-1 trial, which is randomized and which is a very precious data set, of course.
Yeah, that would have also been another question. If you use alpha-NOTA octreotide in conjunction with therapy with lutetium dotatate for the different, let's say, targeting vector and affinity profile, but that you addressed perfectly. Let me ask you one other question. Like, what's your? I was really surprised to know that alpha-NOTA octreotide is already approved in Belgium. So this is really great. And what has been your experience so far in clinical practice? Are you using this in conjunction with gallium dotatate, both tracers, or are you using only alpha-NOTA octreotide, or how do you select which patient becomes which imaging agent?
Yes, it's the latter. We got the approval somewhere in March of this year. Sorry, I don't know the exact date, but quite soon after that, 1 or 2 months later, we started using it in clinical practice. And one of the reasons is that we had, due to the economic crisis in Belgium, we have indexation of salaries in Belgium, so costs have increased very much in our hospital, and we wanted to do cost reduction. And so we did not renew our gallium generators. We still have our old one, but we did not buy a new one. And we started using the aluminum fluoride, and this has been approved by the Federal Agency for Nuclear Control in Belgium based on the data that I have presented. And so since then, we typically now do twice a month 14 patients, so we use 2 PET cameras, and so we produce 1 batch. We inject a little bit less activity, but we keep the same scan time, so we're between 2.4 to 2.7 megabytes per kilo that we inject now. And the image quality has been very good, and I also compare it, we have many patients that we see in follow-up, and so that come repeatedly every 9 to 12 months for a follow-up scan, and so then we have comparison with the gallium, and I don't see any red flags or major differences, and we see still very high SUV max uptakes, and so we've I think I have a PhD student that will do an analysis of our patients that we have scanned at our center. For the moment, I think we are at 149 patients that we scanned using the F18 molecule. I've also seen meningiomas, for instance, that were not in our primary trial with very high uptake, with a max around 40, and that we even want to send for PR. Well, we will do PRT, which is a bit difficult because it's not reimbursed in Belgium for meningiomas. But so we don't see any problems in using it, and we are very pleased. And so we can scan quite a large patients, clearly larger than we did before with the gallium-68. Actually, we did this kind of transition in 2019, if I'm not mistaken, with PSMA as well, because there we could not catch up with gallium-68 PSMA. We had too many patients, and there we switched to fluoride-18 molecule.
Can you also deliver doses to other facilities, like a commercial radiopharmacy, or are there other radiopharmacies, let's say in Belgium or even abroad in other countries in Europe, adapting this?
Yes, so we could deliver to other centers, but for that we first need to get reimbursement in Belgium, because the other center, of course, will be willing to build this, and there we need to make the reimbursement in place. So I'm working on the dossier now, so I hope to submit this by the end of the month of September and in October, and then hopefully in some 6 months' time it will be reimbursed in Belgium. And then other radio pharmacies in Belgium can also produce it if they apply for a permit, and then they can introduce the similar reimbursement dossier, and likelihood that then their version will also be reimbursed. So in Belgium, it's the molecules are reimbursed per production site. So if we make it in Leuven, it's reimbursed, then it's only our molecule that is reimbursed in the system. But others can do a kind of copy-paste of the paperwork and then also get the permit. That's for the Belgium situation, which of course is important for me and patients that I treat. But I've also been contacted by other companies, for instance, from Israel, from Australia, from the United States, that are also interested in this molecule. So I think it is a potential alternative for gallium-68, and I think clearly, as you have done very well in South America, this is something you can distribute. You can make quite a lot of it with the cyclotron, and with the half-life, it is this. You can distribute it. With gallium-68, we have never distributed something in our center. We only had barely sufficient to scan our own patients. With F18, we do distribute molecules such as FDG, fluor, PSMA, FET to other centers. And in the future, we will certainly be interested in doing it as well with the fluorooctreotide, as we have called it, to be able to pronounce it a little bit easier. So yes, I think we will see initiatives as yours in other countries and other parts of the world picking up this molecule.
Yeah, that's. I also think so. I know in Brazil, our colleagues are also working on this, using this already in patients. I can tell you that in Mexico, also at the Autonomous University in Mexico, they are producing alpha-nota-octreotide since 1 or 2 years for clinical use. And we have one guest also connected from Buenos Aires, Argentina, Gustavo Messina, and he asked a question for Noemi. And don't know, Noemi, are you connected? Can you hear us?
Yes, I can hear you.
Wonderful. Hi, Noemi. Also, thank you for your great talk. So, Gustavo, and I think Bakon and the colleagues are also interested in implementing nota-octreotide, and he would like to know if you use a certain quality grade for the sodium chloride for the preparation of the eluent vial for the radiosynthesis.
Yes, thank you for the question. For the sodium chloride, actually we use a saline solution, a 0.9% saline solution, which I believe to be a USP grade. But it is true that in a general way, for preparation of reagents, we always try to select the super pure water and metal trace or trace select grade of reagents when possible, because it's always better to reduce the possibility of metal contamination. And also we started as well to add some ascorbic acid in the precursor solution in case there would be some traces of metal left, to just be prepared in case it would happen.
Perfect. Thank you. Then one more question from my side. I would just like to know from you, or how do you see this? What would be the next steps necessary to get, let's say, a GMP grade cassette and kit for this aluminum fluoride chemistry? I think this would be really helpful to accelerate the adoption of this imaging agent.
Yes, indeed, it would be perfect. I think that it wouldn't be too much work. The main problem, I would say, for now, the main challenge is to simplify the preparation of the reagents, because for now many vials need to be prepared last minute, and it's not very practical for people who want to do a regular production. So we are now working on simplifying the preparation, but I think it's really the last step before we can just launch the product.
Sounds great. So still a little bit of work to do in the lab before we get there.
Yes, indeed.
René, you also had a question regarding a certain indication, right? So I pass this on.
Okay. Thank you. Well, Professor Derus, thank you for your excellent presentation. I have a question. We have observed in some patients with lung carcinoid a lower uptake with fluor nota octreotide versus gallium dotatate. What is your experience with, in this case, patients with lung carcinoid?
Yes, so lung carcinoids are one of the patient populations for primary tumor site where with somatostatin receptor imaging we can have, specifically in the metastatic setting, lower uptake. They are amongst it's the primary organ that we have the most most refusals for PRRT when we scan them to see the expression. We haven't observed the same systematic problem as you had, but we've had some patients where we had some mismatches in some lesions. So I've seen a nice case with the liver lesion that was presented on your experience, but we haven't observed it as a general phenomenon. And I was thinking that it did not pop out in our analysis, but I have to say that in our trial that we did, we don't have that many primary lung cases. So that's gonna check how many it were. I don't know by heart that we have with a primary lung, but we scanned 7 patients. And of course, these are some of those were primary tumors that lit up very well, but that only had the primary tumor. So it's basically open, but it might be that it has to do with the differential expression of the somatostatin receptor. But I do know that it can be sometimes a challenging situation.
Okay, thank you.
Thank you, Christoph. Maybe one question more from my side. For those nuclear medicine centers and physicians who are interested in adopting these new radiopharmaceuticals, we have seen that you image 2 hours post-injection with excellent results, and you get superior detection rates as gallium dotatate. The workflow in the standard nuclear medicine facility is basically injecting, waiting 1 hour, and then proceeding to imaging. What do you think? How important is the incorporation time, and would you recommend to really keep it at 2 hours, or is 1 hour post-injection also okay?
Yes, that's a very good question. So we started replicating the data as we had it. So we stuck to the 2 hours that we used to do our multi-center trial. Of course, we don't have that many patients. We only studied 6 patients, in which we studied at the different time points. At the 1 hour time point, we detected 83% of the lesions, so the union of all lesions seen on all scans, and At 2 hours, this increased to 90%. So we consider this to be a substantial increase. But we are now looking at data from our trial. We look at the clinical impact, and there, of course, we expect that the fact that we see somewhat more lesions on the clinical level does not make that much impact. Of course, if a patient is metastatic and you're going to go for systemic treatment, it doesn't matter if he has 20 or if he has 30 lesions that you see in the liver. And so maybe that once we finalize these data, we will move up the scan a little bit and reduce the uptake time, and maybe go for 1.5 hours, as in somewhere in the middle. But in the beginning, we stuck to replicating as much as possible what we did in the clinical trial. So for the moment, we do it in 2 hours, and we for the moment keep the patient at our department, but we are negotiating with our radio protection officers to have them go out of the department, as we do with patients that come for a bone scan, and then we image after 3 hours, and then come back after 1 hour and 45 minutes, because yes, you need to have more space in your departments to have the patients there. But so I think in scanning at 1 hour, yes, you will get almost the same picture. You might miss some lesions. How important are those to be debated? But in the trial, we wanted to make sure that we put all the let's say that we exploited the potential of the F-18 as much as possible, and that's why we went for the 2 hours, and for the moment we still replicate that.
Great. I think the same is true for PSMA 1007, which we also use here in Chile. You get better images and better contrast if you wait 90, 120 minutes post-injection, but in the end, there's not so much patient in the routine workflow.
Yes, that's very interesting, because we also use 1007, and there we started it, and we did it after 1 hour based on the FDG work, and then we are aware of the publication that showed that we get a better results, and I showed it to my colleague who's responsible for the prostate program, and she said, yes, let's keep it as it is, and things go fine like this, and everybody is satisfied. So yeah, if you're an adopter, you can maybe go somewhat easier. For this, we stick to the 2 hours, but I'm not guaranteeing that we will do this indefinitely.
Let's say it's a good recommendation for new physicians who don't have a lot of experience with this imaging agent to start at 2 hours, and then they can work their way down. So here we have another question from Maria Paz Martinez Vidal from the audience to Arlette. Ella menciona en la publicación, los estudios fueron adquiridos con un tiempo de fijación de 60 minutos aproximadamente. ¿Cuál es el motivo? Y por otra parte, como médico nuclear, considera que los estudios pueden servir para el seguimiento de pacientes con tumores neuroendocrinos, donde ya se utilizó galiodotatate y después se sigue con FAN o alfa-nucleotide.
Claro, la misma incógnita que tenemos nosotros como centro, ¿cómo comparamos la captación del galio dotatate versus el fluoroctotide? La verdad que son trazadores distintos, la intensidad de captación no es comparable y no hay estudios todavía que nos puedan asegurar cómo responde este fármaco a la terapia, o sea, cómo evaluamos una respuesta a terapia solamente con el fluoroctotide. O sea, eso todavía está en estudio, no lo podemos asegurar, no sabemos cómo aún evaluar la respuesta a tratamiento, por ejemplo, con el fluoroctotide. Actualmente solo nos estamos enfocando en si las lesiones siguen captando o no captan prácticamente. Pero en intensidad de captación, ambos radiofármacos creo que no son comparables. Desde mi punto de vista como médico nuclear. No sé si existen más estudios, pero la verdad creo que no han salido.
Perfecto. Bueno.
Y la pregunta de los 60 minutos...
¿Cuál es el motivo de la incorporación de 60 minutos?
Creo que en el momento de estos pacientes que se hicieron entre el 2020 y el 2021, todavía no teníamos los antecedentes de que a las 2 horas se podían evidenciar más lesiones, me parece.
Nos anticipamos un poco el estudio
en el estudio, sí.
Los colegas de Bélgica. Bueno, entonces yo creo que no tenemos más preguntas por el chat. También agradezco a todos los presentadores, Christoph, Noemi, Arlette, muchas gracias por la participación. Christoph, we will see each other at the ENM. So let's have a chat about possible collaborations. I think we will have a lot of data here from Chile. We can contribute to joint studies we can do together. So I think we should explore this. And aquí surgió otra pregunta, ¿no? Eso. Esteban de Incan, perfecto. ¿Cuándo creen que IVA ya cuente con kits para síntesis del ALF para la cintera V2? Bueno, perfecto. Es cierto que estamos en contacto con ustedes y otros centros también en México que están esperando esto, y fue un poco la pregunta que hice a Noemi. La idea nuestra en conjunto con IVA es de finalizar en el fondo el diseño del kit y del proceso de la síntesis. Y aquí todavía quedan algunas mejoras por hacer, porque nosotros obtenemos resultados muy estables, muy buenos, pero la preparación del kit en sí todavía es un proceso bastante complejo. Estamos buscando simplificarlo un poco para reducir los reactivos, los insumos necesarios para la producción. Entonces todavía nos falta como este diseño final del cassette para avanzar con la generación de kits GMP, pero seguro que va a estar y pronto. Bueno, con eso agradezco a todos y cerramos el webinar aquí. Muchas gracias por la participación y les avisamos por próximos eventos que hacemos en el futuro. Muchas gracias.